Okay, so let's go ahead. Hi, can you hear me?
Good morning, I can.
Okay, good, good. We'll wait a minute or two in case anyone else shows up. So
Sounds
we're
good.
going to Monday, not for you for the exam. We're going to do another day.
Actually, I was not able to go on vacation, so I can do it Friday. Yep.
Okay, so I have to change that. I got to write that down or I'll screw up.
Okay. And then did you get my emails about the atherosclerosis and hypertension discussions?
I did not check emails yet this morning. I worked on some of the discussions last night. So there might be grades posted for hypertension and I think for some of the atherosclerosis ones, but I'm not done with all of them.
Okay,
So,
I just wanted to make sure you had mine.
I think so. Sometimes if you submit stuff late, I have to do a kind of a circular thing, kind of a roundabout way to grade it.
I gotcha.
So usually it's okay.
Okay, sounds good.
All right, so we have an exam on Friday and Saturday. So it'll open Friday, close Saturday night. It's going to cover chapter 16 on the reproductive system. I got to look here. 16 is reproductive system, 19 is inheritance, and then 20 is DNA technology. And we're working on evolution, but it's not going to be on the exam. Okay, it'll be on the final exam. So a lot of that exam material is again going to come from the quizzes. So you should look over your old quizzes. It's not that I'm going to pull. I'm trying to, review the same things that the quizzes reviewed. You might get different questions because there's always a pool of questions. So it won't be exactly the same, but it'll be similar.
Okay.
So in the reproductive system, we're talking about the male and female reproductive system, and we kind of emphasized that it's that there's a control system that involves the brain and some hormones. They talked a lot about birth control and about sexually transmitted infections, and I think we had a forum on sexually transmitted infections. So that was the first of the 3 chapters. Let's see if there's anything else. Talked about sexual response, lots of stuff on the different types of contraception and birth control. And I had a little thing on one of the Zoom discussions where we were I talked about success rates and things like that. Oh, and then yes, that's the forum. So the forum was on cervical cancer and the vaccination against the infection, and so we did that. Okay. Then the next chapter was chapter 19 on genetics and inheritance. So this is kind of related to the reproductive system because it's what you pass on. And so children are, the sperm and the egg contribute 50% each for the genetic material. So half of your genes come from your father and half from your mother. But what half you get is random. Okay, so that if you have a brother or sister, they can look quite different depending on what they, the genes that they got. And then we have, for every trait like eye color, we have 2 genes, one from our mother, one from our father. Okay, those genes don't mix, they're separate. And the physical trait that we get is a result of which gene is dominant. And so some dominant genes always show up. Like if you have the gene for brown eyes, you will have brown eyes. Okay, there are also recessive genes. And the recessive genes can be hidden by dominant genes. And so like my mother had brown eyes. I have blue eyes. And so my mother didn't give me a gene for brown eyes. She gave me a gene for blue eyes. And so did my father, who had blue eyes. And so that's, you can have these hidden genes. So my mother had a hidden gene for blue eyes. Okay. She didn't show that trait at all, but she passed it on, and I show it because I have 2 of those copies. That was really confusing because, you know, they couldn't figure out inheritance for the longest time. Okay. And so that's the confusing part. And there's lots of recessive genes that are mostly hidden. Lots of diseases like sickle cell anemia, Tay-Sachs, muscular dystrophy. Those genes are caused or those diseases are caused by recessive genes. And
Yep.
those recessive genes, the parents then are healthy, and both parents carry the gene, and they can pass it to a child. Okay, let's see what else there. So now with genetic testing, we can identify those genes. We can figure out where they're going. What are the chances that you're likely to pass on a recessive gene? And so that was our big thing, working on that. They also talked in there about the sex chromosomes. And, you know, I don't talk about it much, but this is a really big issue. They talked about the types of genetic mistakes that happen during the production of sperm and egg. And so on page 457, they talk about alteration of sex chromosomes. And this is a big topic now because people are talking about transgender this and transgender that. And you have some people saying there's only men and there's only women, be one, you know, be what you're born. And it turns out that in some cases, you know, they're not a man and they're not a woman. They're something in between. Okay, and so you can get abnormal numbers of sex chromosomes. And so they go through XYY, XXY, XXX, these abnormal numbers of chromosomes. Each one of these is about as common as Down syndrome. Okay, each one of them. So that doesn't mean that there's hundreds of them, but that means, you know, in Freeport we have 20,000 people, 25,000. Okay, in about 1 in 500 births, you have a Down syndrome's birth. Okay, each of these kind of misalignment of sex chromosomes is about a 1 in 500 births. And so in Freeport, there's maybe 40 or 50 of these people. Okay, that they're not what we would say truly men or truly women. They have some sort of intermediate combination of the characteristics. Those are hard lives, like Down syndrome. Those are hard lives, and we can be thankful that it's not us, as long as it is not us, but it's one of those things. And so that's not to excuse everyone's behavior, but it's to say these intermediate characteristics are fairly common. And so, okay. All right. And then last week we had the election, so we missed on Tuesday. Monday, nobody showed up for discussion. Yeah. And so that happens. I understand, they're voluntary. So I really didn't get a chance to talk to anybody about the DNA technologies. So I'm going to share the thing and ask questions if you have one. So let's see if I can find the PowerPoint, and I'm going to share it if I can.
All righty.
Okay. So there should be a screen where it says DNA technology and a bunch of slides. Do you see that?
Yes, I do.
Okay. So these techniques are kind of new. They were coming around when I was becoming very common when I was a grad student. So that's like 20 years ago, 30 years ago. And they have various names, things like that. So you see that technology, how to cut and paste with DNA?
Yep.
Okay. So when they figured out what DNA was and what it was, you know, that it was in all cells and that it was a genetic code, they started figuring out, well, DNA has to be copied and the cell has to deal with DNA. And so they realized that cells must have enzymes that are able to do things with DNA, like to copy it or to cut it or to repair it. And so biologists started looking for these enzymes. And they found lots of them. And these enzymes allowed you, if you could make the enzyme, you could do specific things with DNA. And so some of the really important ones could cut DNA at specific sites. These are called restriction enzymes. Okay, and other enzymes acted to glue DNA back together. And so as biologists and chemists got more and more familiar with these enzymes, they realized we can cut and paste DNA. We can cut DNA out of one cell and paste it into another. Okay, and it's improved even more. Our book doesn't have, there's a new technology called CRISPR. Our book doesn't really have anything about it. But when they talk about DNA and DNA technology, genetic engineering, they're really talking about using these enzymes to manipulate DNA. And then you can put DNA in a cell and let the cell grow. And so you can take DNA from one organism, cut it out, and use an enzyme to put it into another organism. This has been really wild. It's been allowed us to do amazing things. And we can take DNA that we want, and we can put it in something called a plasmid, and then the plasmid is designed to go into another cell. And so we can take DNA and transfer it from one cell to another. If we have a gene that we know there's something good there, we can get the cell to make the gene. And this has allowed us to produce lots of different types of medicines. If you have a deficiency of a hormone, we can now make the hormone. So like if your pituitary gland has been injured and you don't make growth hormone, the doctor can find out for particularly small children and say, okay, your pituitary gland isn't making this hormone. Here, we can give you it. We've made it and we can give it to you and it's safe. This has been most important for people who have diabetes. Untreated diabetes is fatal, 100%. 120 years ago, everyone that got diabetes died. And it would take 2 or 3 months, but they all died. Well, now we can make insulin. Okay, and when they first figured out that they were missing insulin, what they had to do was take insulin. They would take a pig that went to the slaughterhouse and they would take the pancreas out, and then they would grind it up and purify insulin. And it took many, many steps, and it wasn't always as pure as you would like, and it was very expensive. Well, after this technique, they were able to take the gene from a human cell, pancreas cell, put it into a yeast cell, and they can grow that yeast, and the yeast makes insulin. Okay, and so you can just keep feeding the yeast sugar, and the yeast keeps making insulin. So you get a much higher quality medicine, much cheaper, and it's much easier to purify. And so this has really been important. And so we make lots of medicines this way, particularly hormonal ones, but others as well. We make vaccines these ways. And even if you watch the movie, even a bigger impact has been in crops, in agriculture, is we've been able to take genes that make toxins that target insects and so put these into corn and soybeans and other plants. And these things, that corn now makes its own insecticide. So this corn, the yield goes way up. The farmer doesn't have to spray as many chemicals on the plant. He doesn't have to use the tractor as much. There's all these advantages. And now the dominant seeds in corn and soybean and cotton and potatoes are all genetically engineered. And we can do this for a variety of things. But it's basically cutting a gene out of somewhere that we like and pasting it into somewhere else and learning how to grow it. It's not a hard technique. We do it in labs with first-year students in biology. It involves using a pipette. So, and really, it's easy. Yeah, it's using, you think, oh my God, it's genetic engineering, and it's really pipetting. You take the enzyme, you move it to the DNA, and then you mix the DNA with the other DNA. It's very easy. And so it's just finding the right gene, getting it into the right organism. But it's been very, very successful. Okay, another technique that's really been helpful in another way is we were able to copy DNA. And this is called the polymerase chain reaction. And this is using an enzyme that copies DNA from bacteria that like growing in hot springs. And so this enzyme is called Taq polymerase. It's from this hot springs bacteria, Thermus aquaticus. And if you have a sample DNA that you want to examine, but you have a very small amount, you take that small amount and you put it in a test tube with the raw materials for making DNA, primers and nucleotides, and the enzyme. And this thing then goes into a thermocycler that heats it up high enough so that the DNA strands separate, and then cools it down to a temperature where the enzyme works. The enzyme attaches to the DNA and copies one of the old strands, and it keeps copying, and it goes through these cycles. And so this is cheap. Again, it's cheap. And you can take DNA from one cell and make unlimited amounts. Okay.
Was that a test question or a quiz question last week?
I believe it was.
Okay, I got that wrong. What was the answer? How much of a cell do you need to copy, or DNA do you need?
One copy. Just one copy of the DNA is what we need.
Gotcha.
And so I have a bunch of funny stories about this. One is when this reaction became available when I was in grad school. And I was like, oh my gosh. And you could buy the enzyme, and the enzyme would cost like $30,000. And then you could buy this machine that would do these temperature cycles. And the machine cost $150,000 when I was in grad school. Okay. I have one now in the lab at Highland. Okay. The machine has this fancy name, thermocycler. And so when it was new, they charged whatever they could get for it. But all it is really a hot plate. It's a hot plate that cycles between these two temperatures. And so they were charging $150,000 for a hot plate.
Wow.
But this has been great because you can take a really small sample and you can get unlimited amounts of DNA. This has been very, very bad news for criminals.
Yeah, I bet.
Because now if they leave a single hair, they can be captured because their DNA is unique and the hair contains enough DNA. In the early 90s, there was a case where there was a murder case where a man was killed and they had no suspects. But when they found his body, they found cigarette butts next to it that the murderer discarded a cigarette butt. And they were able to look at the cigarette butt and get a skin cell off of it. And they identified the DNA, and luckily it was someone that had been previously convicted of a violent crime, and their DNA was on file. And so they went to the person's house and they found more evidence, and it helped them solve the case. So you don't want to commit a crime anymore because it is impossible to go somewhere and not leave a hair and not leave a skin cell. And so violent crimes were much better at solving them, and we're much better at finding mistakes in justice than we used to. And I think the forum was about that. So So really good stuff. Okay, let's see what else. Okay, and so often this is combined with DNA sequencing, where you sequence the DNA. Again, this is using an enzyme that builds new DNA, but we have a way of finding out what is the last nucleotide. And so we terminate the sequence at various spots with a fluorescent dye, and we can tell which fluorescent dye ended the sequence. And so a computer assembled these fragments together to analyze them in sequences. So we've sequenced the whole human genome. We've sequenced the genome of corn. We've sequenced the genome of all the organisms we're interested in. And now we can sequence genomes relatively cheaply. The first one cost $1 billion. Okay, now you can get it done at the hospital for a couple thousand. And so relatively cheap. These technologies, again, can find rare genetic diseases that we just could not identify before. And now we can find them pretty easily. Okay, and so these are automated, and this just shows the sequencer. And again, it's a machine. And so this is still more expensive. I couldn't have this in the lab, but it's not hard to run. You load the enzyme in, you load the raw materials in, and your DNA that you want to sequence, you turn the machine on and it runs cycles, and out comes a sequence of labeled antibodies that are identified by a computer. And so basically the technician loads this in the morning, drinks coffee for a couple hours, and then looks at the results. And
That
so
sounds like a pretty easy job.
it's a good job. Yes, good work. And it's you know, this was a lot to find, but once it's fine, once we've identified the technologies, they can be automated, and so they work well.
Mhm.
Okay. We also do something called DNA fingerprinting that doesn't sequence the whole genome, but just these really variable sequences that are usually unique to people. And so we cut the DNA into pieces, and we can sequence the pieces or just separate them on a gel, and then we look for matches. And I think the Innocence Project, that was the forum, was looking at some of the
Yeah,
stuff
that
there.
was really eye-opening for sure.
Right, and so we find that in previous convictions for particularly murder, about 5% of them, we can clear them. We can say absolutely for sure this was not the person that did this. Even though that the blood type was the same, the murderer and this person that was convicted had the same blood type, the DNA is clearly different.
Mhm.
And the forum really didn't ask you to, or didn't explain everything. But in a lot of cases, not only does it clear one person, but it points police to the direction of another person. And so, you know, we often hear like politicians talk about crime a lot, and that crime is rising, and the truth is it's going down. It's going down dramatically over the last 40 years because of techniques like this, because of things like cell phones and easy availability of internet cameras to monitor, you know, buildings and things like that. You know, I was where you are in the 1980s, and I had a friend that worked at a gas station, and he got a job in the evening at a gas station, and it paid really well. He was working third shift and making, you know, several times minimum wage. And he was very excited to get the job. But what he didn't realize is that the gas station was right on the expressway that was not too far from a big city. And so every weekend, people would drive out from the major city and rob this gas station very quickly and get back on the expressway and go back downtown. And so that about once every 2 to 3 weeks, the station was robbed at gunpoint.
Oh
And so
my gosh.
yeah, but that just does not happen anymore. My daughter
No.
just finished college working at a gas station, a Quick Trip, and she said, "Dad, we've got like 80 cameras. We've got
Yeah.
80 cameras. Nobody could rob us without getting caught. They get caught. We get the license plate of every car that goes through. There's no chance that someone would not get caught." And so 40 years ago, when I was in getting out of high school and going to college, people were doing that kind of stuff every weekend. Now those people are caught right away. And same with murders and rapes. There's a much greater chance that we're going to find those people fast. And so it's not that there's no crime, but that it's much harder to get away with it and have a sequence of crimes. It's much more likely we're going to catch someone. And so that's DNA fingerprinting and DNA for using identification in crime. It's a really good thing. It's not only helping criminals, but it's helping innocent people. And when this first started, I've got in Illinois here, our governor, there were between 1977 and 2000, there were 12 executions. And the governor we had then, when this technology came online, immediately said, you know, we should do this. And within a 4-year span, they had 13 convictions overturned. And he
That's
realized,
crazy.
oh my God, we've got to stop executions,
Yeah.
because a significant number of them we're executing people who are not guilty. And so that's how the death penalty stopped in Illinois, is because of that. And so it's amazing sometimes what new technology can do, that it can help us. Not that it's not without any problems, not that
Mhm.
there's not the potential for privacy invasion or someone discriminating against you based for insurance or something like that. There are problems. But the technology has allowed us to do things and see things in new ways that have been very, very helpful. Okay, I think there's another slide. Okay, yeah, let's see. So then I go into safety. Gene therapy, I'm not going to say too much about it. That's overblown. There's a lot of people that are concerned about safety. I think, you know, the Franken-food thing is overblown.
Yeah.
Food that's made from genetically engineered crops, that's not going to genetically engineer you. You're going to cook that food and eat it. It just has genes that came from particular plants that help the plant. And so you're not going to be genetically engineered. That's not something we ever need to worry about. But there are issues about who owns the genes. You know, like the idea of when we introduce something like the PCR technology, that it costs. How much does it cost? Who owns the genes for like the BRCA gene? It's a breast cancer gene. The testing is extremely expensive because they want to make money on finding that. They don't really own the gene, and the test. I have problems with making that so expensive that not everyone can afford it. So anyway, okay. So let's end that. Do you have any questions? Did that help at all?
Yeah, no, it was really, yeah, it was good. I think you covered most everything. I don't have any questions.
Okay. Okay. All right. So we're starting on evolution for the chapter on evolution. I just kind of the general principles are important that because DNA can change, organisms can change over time, and those changes can be passed on. And if they're good changes, it's likely the organism is more likely to leave offspring. Okay, evolution's, we know the genetic mechanisms. It's not a mystery how it happens. We know that everybody has this huge set of genes. Those genes change because there are mistakes made, and sometimes the mistakes are bad, but sometimes they're good. Okay, and sometimes a mistake might be good and bad in one situation, but good in another. And so genes not only can be labeled good and bad, but they should be labeled as good here, bad there, and things like that. So so we know that certain genes give people certain advantages, and that over time genetics can change. And so it's not a stretch to think one species can change into another. And we have lots of examples of genetic change happening that we can measure, that populations change. Particularly small populations changed. If you have a small population in there, they can't breed with newcomers, that population will change genetically from other populations. And we see that on islands. Okay, we also see that in communities, like in Amish communities, because they don't mix with other people. We see the genetics of Amish communities, like in Pennsylvania or Iowa, is very different from surrounding communities. And because those started from small communities, they have lots of issues with genetic disease because they're relatively inbred. And I think the forum, I just opened the forum this week. It's on Neanderthals and using the genetic technologies that we just talked about to compare the DNA of Neanderthals and people and their similarities there. So, okay, this won't be on the test, the exam 4, it'll be on the final. Okay. All
and
right.
what chapter number is this?
This is 22, Evolution and the Origin of Life. And if you look at the hints, the chapter hints, it says focus on certain chapters. We're not so much on the evolution of life, general principles and evolution of human and human relatives.
Okay.
All right. So I think if you don't have any more questions, I think I'm done.
Yeah, I think you covered it all for sure.
Okay, and I've written down that you're going to take the exam with everybody else. If that changes, it's okay. Just send me a note. Okay.
Okay, sounds like a plan.
Okay. All right. I'll see you. So
All right.
good luck. Study hard.
Thank you. We'll see you later.
Okay. Bye-bye.